development of the Drugit platform you'll hear second about today. So now I'm going to explain how this session is going to work. So each team will talk for around 20 minutes. And then each talk will be followed by 5 to 10 minutes for questions. And you can ask questions either by using the Zoom reactions to put up your hand or by entering questions into the chat. I would like you to ask to keep your microphones muted unless you are asking a question. Now just let me introduce the first team. So our first team is joining us from Brazil today. They predicted more confirmed hits in the cash challenge than any other team. And today, 3 members are going to take the stage in turn. First, we have Karina Machado, Associate Professor at the Federal University of Rio Grande. And she will take the stage to detail the methodologies used in rounds 1 and 2 of the cash challenge and highlight the tools and algorithms applied. Following that, she will focus on the consensus score and the filters used. And second, we will have Federico Kramer, also an associate professor at the Federal University of Pelotas, and he will discuss the application of machine learning, emphasizing the filtering process from the data available at Enamine, and the use of deep learning for predicting protein ligand scores and the similarity-based search for molecules. And finally, Adriano Verli, also an associate professor at the Federal University of Rio Grande, will present the details of the docking simulations he employed in rounds 1 and 2, including receptor preparation, binding site definition, and execution in HPC environments. Please, Karina, take it away.
Hello, everybody. I'm going to share my presentation. Okay, just a second. Hello, good afternoon. I'm Karina, and today I'm going to present a little bit about our strategy for participating of Cash Challenge 2. It's a pleasure to be here. I'm very happy to present our strategy and together with my colleagues talk about our work at the university. First of all, I would like to show you where we are. We are in the south of Brazil. This is a map of the Brazil. And we are in a state called Rio Grande do Sul. So we can see that we are in the south and in 2 cities that's very close actually, in Rio Grande and Pelotas, where Rio Grande are M and Adriano, we are now, and Pelotas, Frederico is talking from. So I divided our presentation and explain you how was our strategy during the hit identification. We divided it in 4 stages, and then how was our strategy for merged selections, and after how was our strategy in hit optimization. For each of these parts of our strategy, we are going to, after my brief presentation of overall strategy, we are going to detail some parts. This stars with the letters indicates that some activities are going to be detailed by each one of us. I'm going to detail about how we did the consensus score using our in-house scoring function or FL score. After that, Frederico is going to detail about 3 parts, the database diverse, the representative molecules we consider in docking, and about our target-specific scoring function. And finally, Adriano is going to detail about how we prepared the molecule for the docking and how we executed these docking simulations in a cluster environment. So in the first step of our strategy in the stage 1 of hit identification, we started using the molecules from Enamine. And in this database, we choose the in-stock zinc 20. We choose this database of Enamine because the molecules in this database are already in 3D format. That is important for us for docking simulations. Then, considering these molecules, it was around 4 million molecules. We computed the Morgan fingerprints using RDKit and Bamboo. And after that, we clustered these structures using a hierarchical clustering algorithm. And after that, just because I'm in the front, we obtained around 12,400 molecules that we consider was a database of diverse molecules with a difference between them. After that, in the stage 2 of hit identification, we prepare all these molecules for docking simulations. We prepare the target protein, the NSP13, and then we performed I closed the, no? Okay, sorry. And then we performed the docking simulations in a cluster environment, and we generated the 12,000 docking results. In the stage 3 of hit identification, using the docking results, we generated a target-specific scoring function for the NSP13 target. So, considering as predictive attributes for our scoring functions, we consider the Morgan fingerprints of all the 12,000 molecules, and as target attribute, we consider the VINA score computed by our docking simulations. So having this scoring function target specific, we submitted all the molecules presented in Enamine, and we selected the most promising molecules according to our target specific scoring function. This was a total of 1,000 of molecules. So we performed again docking simulations using the same environment. The NI script generated using an in-house framework for virtual screening. And then from each of these 1,000 docking simulations, we extract only the best docking position of the ligands and the score with VINA. And we also computed 4 more other scores considering different scoring functions, including our in-house scoring functions or FL score. So having in hands all these 5 scores, we combined these results using a consensus. And finally, we applied the defined filters defined by the organizers of Cache Challenge 2 in these molecules. We decided to apply the filters only after the consensus and our final ranking, because sometimes the values of the descriptors that was related with the filters was really close to the limits. And if the molecule has, for example, molecular weight very close to the limit, but was good position in our ranking, we maintain the molecule, okay? So in the next stage of the Cache Challenge 2, during the merged selections, we started with the 2,200 molecules, the top 100 molecules of each group. These molecules were provided by the organizers. And then for each of these molecules, we obtained generated the 3D structures. We optimized these structures because in that case, we just had the SMILES format of the molecules. And then we performed the 2,200 docking simulations. We computed all the 5 scores, and then we combined these scores using a consensus, as we have done for the other steps. However, in this step, we needed to indicate also if the molecules was potent, weak, or inactive according to our strategy. So in that case, we applied a little bit different consensus, because we discretized all the scores between potent, weak, and inactive, okay? Finally, in the last step of hit optimization, We started with the 8 molecules that was hit results of according to our strategy, RFL Bamboo. And then for each of these structures, we come back to an amine and selected from 50 to 100 similar structures using the Tanimoto similarity. And then this totalized about 559 molecules. And then we performed doc simulations. We computed the 5 scores. We applied the same consensus score. We applied the filters as the previous step. And finally, we generated the final 42 molecules that we indicated in this step. Now I'm going to detail about the consensus score. So as consensus score, as I have mentioned before, we are considering 5 different scoring functions. It's important to mention that all these scoring functions, they are quite similar. They are based, they are or machine learning based or empirical, and they are using a little bit similar molecular descriptors. So they are not quite different, okay? And then for combining these results, we used a simple weighted average, okay? And then we applied the filters, for example, the molecular weight, log P, that was defined by the organizers. And for the consensus score in the merged selection step, we needed to discretize each one of these scores. We discretized between potent, weak, and active. And to combine these results, these classes, we used a voting. Like a voting, if, for example, according to 3, 4 scoring functions, the molecule was indicated as potent, it was present in our list. If it was indicated most of the scores functions as inactive, this was removed from our list. And finally, I would like just to highlight that we used during all the consensus our in-house RFL score. This is a machine learning based scoring function that we have been working since 2018. And this scoring function we are using to train the score function, the random forest algorithm, and to select the most important features, the lasso algorithm. And it's important to say that we are using in our scoring function around 722 different attributes related with the ligands, the receptors, or related with the complex. Now I pass to Frederico. He's going to present about the next step, the representative molecules for docking.
So based on the initial data set from Enamine, we computed the Morgan fingerprints using RDKit and Bamboo, which is a XR modeling pipeline we have developed at my laboratory. So based on this fingerprints, we performed a random subsampling because just to reduce the number of molecules and allow us to perform a fitting of a PCA model. So we reduced it to something about 132,000 molecules. And then we fitted a PCA model, a principal component analysis model, that preserved 95% of the variance of the data. And then we used this same model to perform a denoising of the fingerprints, because as the fingerprints are binary, probably some bits in these fingerprints would be very hard and very rare. And then we wanted to reduce the effect of this kind of noise, low frequency bits in the further steps. So based on this denoised fingerprints, we then performed dimensionality reduction using UMAP, and it reduced the feature space to just 3 components. And then we used a hierarchical density-based clustering algorithm, which is HDB-scan, to cluster the molecules in approximately 190 1,140 clusters with molecules, at least 10 molecules, with at least 10 molecules. And then based on these clusters, we selected till 100 molecules or less, depending on the size of the clusters. And based on this molecule selected, we performed then the docking steps. So next slide, please. Then after the docking, we scale the energies and to be between 0 and 1. And then we used the noise and fingerprints to train a stacked neural network with a regressor that is based on a more traditional machine learning model. This traditional machine learning model, which could be a random forest or extremely randomized machine learning classification model or gradient boosting regressor, was trained using Flamel, which is AutoML framework from Microsoft. So there is this neural network that produces a prediction, and then this prediction is concatenated with the denoised filters, and then we recalculate the energy and trying to improve the prediction from the neural network. So in the next slide, we have the architecture of the network. It's a very simple, fully connected, dense neural network. It receives the fingerprints and then outputs the scale energy, and then this energy is concatenated with the fingerprints for the next model. In case of Flamel, we selected LightGBM as the best regressor. So it is used to then calculate an approximation of the docking energy based on the fingerprints. And in the next slide, we have the plot with the correlation between the docking energies from the test data set, which is we separated a part for training and a part for test. And here you have the correlation, the Pearson R, the Spearman R, the R2 metrics, and the mean average percentile error from this modeling in the test data set. In the next slide, we have then the similarity search. After the stage 1 compounds, we performed just a similarity search using RDKit based on the Morgan fingerprints. Then we used Tanimoto similarity to select from 50 to 100 most similar hits for each compound. And then we ranked them and passed it to the next step. And now Adriano will present the preparation of the binding site.
Yes, thank you, Frederico. For preparing the binding site, we started really getting the proteins and the amino acids that were presented in the announcement of Cache Challenge. And we superimposed all these structures in PyMol and highlighted the amino acids that are found in ATP sites, central channel, nucleotide site, and 5-RNA sites. And by superimposing them and inspecting them with many different views and configurations, we decided to select the 5-RLZ protein chain A as our protein to perform the dockings. Having this defined, we go to the AutoDock tools to define which will be our search box. Next slide, please. And this is just 2 figures of the AutoDock tools where we plotted only the cartoon of the 5-RLZ protein and all the amino acids that were listed as interacting. We plotted as these gray spheres, and then we decided we adjusted this box as the search box for all the docking simulations. And next slide, please. And for running, well, then we are facing the situation now where we have a large number of molecules, more than 12,000 molecules, and we have a big docking box. It's almost a blind docs. Then for performing the simulations, we used a framework that we have been developing for a long time now. We have a screenshot of it here, our VS framework. Next slide, please. And in this framework, it makes much easier to run a bunch of docking simulations using Vina, because we can just select all the parameters in this tool. And then next slide, please. And then we can define the box, exhaustiveness and everything. And then next slide, I can show you that what we obtain for our in-house framework is a Python script that runs all the simulations that we have programmed there. In this case, we discovered that our framework is not prepared to run in HPC environment, so we had to do a little trick of running 4 different simulations, because the computer we have here to run these things, we have 4 computing nodes, and we could not submit just one job. We had to submit 4 separate jobs, but we could get all the results in about 9 hours. So this was good that we could get all the results really fast. Next slide, please. And that's it, really. We run all these methods that we just presented for you, and we end up with this in this green box with the 8 molecules that we presented, and this little number on the side of each of these molecules is the number of analogous molecules that we also presented together. And then out of this 46 molecules, we could find 8 that were really interacting. Next slide, please. And here I present our teams. To the left is my team that I lead together with Karina in the Federal University of Rio Grande. And to the right is Frederico's team that he leads at UFPEL, really close here. And in the next slide, we have our contacts. If you want to contact us, you can try by email, Instagram, not Twitter right now, because it's not working in Brazil. Thank you very much.
Thank you, Karina, Frederico, and Adriano, for your nice talk. Are there any questions from the audience? Well, not right now. I give you some time to think about questions. I would have a very specific question to Karina. The bad apple filter used when consensus scoring, what is it exactly? I've never heard it before.
Oh, sorry, I didn't listen. Okay.
Right. So the bad apple filter you used for consensus scoring, what is it exactly?
It's not the name, it's not the filter, it's a website that the organizers asked us to use. It's not,
Right.
I forgot to put what exactly it means. It's just the name of the website. Is something
Right.
that challenge organizers asked us to use to verify if the molecule, some characteristic that I don't remember now, but I just put the name of this server that we evaluated the characteristic.
We do have a question from Mathieu Shapira. Did the confirmed hits score well with the RFL score?
I didn't see the question. I don't know if
I can repeat the question. Did the confirmed hits score well with the RFL score?
I didn't come back to the scores to verify if how was the position of the confirmed scores according to our score. So I didn't come back of the RFL scores to verify if the 8 hits molecules are in the top or where they are. Sorry, I don't have this answer because we didn't verify it yet. Maybe for the paper.
Right. We have another question from Shubhangi. So thank you for the talk. And what site was targeted for docking? Was it the central tunnel, the ATP binding site, or the 5' RNA site?
Yeah, it was ATP site, central channel, nucleotide site, and 5 RNA site. All of them were targeted for docking. That's why we end up with this really big search box. Yeah.
Right, thank you. And Mathieu is asking, were all the confirmed hits docked in the same pocket?
Yes, we consider for all the docking simulations the same pocket. The pocket that Adriano showed, it's a big pocket where we are considering the region where RNA or ATP and the center pocket, every amino acid that was indicated by the papers are part of the pocket. So, but we didn't analyze it after the docking simulations where exactly each molecule has the best score. So I don't have exactly this answer about the pocket. If they
and he's asking whether the protein was static or dynamic.
Yeah, the protein was static. Yeah.
Right.
We thought about considering at least the binding siting as flexible or performing the simulation of the protein and then choose some conformations of from these molecular dynamics, but in fact we didn't do that. We just use it as a not flexible.
It is.
I like to do flexible docking or ensemble docking, but in that case we didn't. The protein was static.
Mathieu is saying thank you and congrats. I would have one last question for all of you. What was the biggest challenge actually in the way you tackled the problem? Would you do it differently now?
Good question. I think for me the biggest challenge was to at least in the hit identification we have no idea if the molecules that we had indicated was okay or not. So we are now really happy that it really worked well, but at the beginning we have no idea. So this was a big challenge. We talked a lot about our strategy, organized it, but we didn't know if it is going to work. So this was a challenge. I don't know what.
I would try to run all the dockings with all proteins. We just decided to use only one, and we used only one in static, but it would be nice to maybe run everything with all the proteins, but we didn't have time. At that time, we didn't have computer power to do that.
Right, thank you.
For the similarity search, I would try to use more modern methods, for example, using Mal2Vec to represent the molecules and then perform similarity search using cosinus similarity. But Tanimoto is a traditional method and it worked well. So sometimes it's nice to keep it simple and use straightforward methods. And I think I would not change because the results were good. So I cannot, but maybe in the next time we can try more other methods.
Right, thank you. Mathieu is asking whether the scoring model was trained. Well, he's saying that the scoring model was trained on a small library, and what was exactly the rationale for this?
You mean the score, the target specific scoring function we trained here for the NSP13?
He's saying yes.
Yeah, it was trained with the 12,400 docking results with a small library. Yeah.
Right. Well then, thank you again. And we move to the second speaker now, Rocco Moretti. So our next speaker is Rocco Moretti, research professor in the Department of Chemistry at Vanderbilt University. Rocco and the Drugit team focus on citizen-aided drug design, and the Drugit platform is a modification of the famous Foldit platform that enables citizen scientists to design small molecules through an online gaming platform. And please welcome Rocco, who will detail how Drugit combines open source software, citizen science, and expert guidance. Rocco, the stage is yours.
Thank you. See, sure. Yes. So yes, as Yvonne mentioned, the approach that we took in the CASH challenge was to use the Foldit platform to crowdsource this sort of drug design challenge with citizen scientists. And so the Drugit platform is, excuse me. Yes. So yeah, so the Foldit platform and the Drugit platform is built off of a core of Rosetta. And so Rosetta was initially designed as a biomolecular modeling platform, specifically for protein folding. But over the years and the 100 labs that are now working on it, it's been endowed with a bunch of additional tools. And so it's not just a protein modeling tool. It allows you to model other biomolecular molecules such as RNA and DNA, carbohydrates, non-canonical amino acids. And specifically for our purposes, there's a ligand docking and ligand modeling component of Rosetta. And so Rosetta's aspect within Foldit comes specifically for through the concept of Rosetta at Home. So Rosetta at Home was built in the early 2000s as a distributed computing way of running Rosetta. So this Rosetta at Home is a screensaver program that runs on your computer while your computer is idle, using those spare CPUs to do biochemical calculations. And as a sort of an added benefit, there's this screen that pops up that shows what the Rosetta is doing on the automatic process under the hood. But one of the early feedbacks the Baker Lab got from the Rosetta at Home project was the fact that people were looking at what Rosetta was doing and thought, I could do better than that. And so David Baker collaborated with Zoran Popovic at the University of Washington to come up with the Foldit platform. And so this takes Rosetta and creates a game environment on top of it. So now game players, just regular citizen scientists, or regular citizens can download Foldit and now play a game where they have easy-to-use tools that allow them to do biomolecular modeling and solve some of these real scientific problems using a game environment. So initially, this was for protein folding and protein design, but the Myler Labs looked at this and said that since we have the small molecule modeling tools within Rosetta, we can extend the Foldit interface to do drug design. And this is what is now Drugit, where we have a game interface where we have a whole bunch of tools that are available, and players can now manipulate the small molecule within the binding pocket of the protein to come up with a better binding structure and a better chemical identity that will bind to this protein. And one of the main ways they sort of explore chemical space is by these 3D modeling tools. So we have a small molecule design panel, which allows players to add additional atoms, add additional bonds, to delete atoms, to change bonds. And we also allow them to add various fragments, such as aromatic rings and other such functional groups. So this allows for free design of the small molecule. But one of the challenges that comes up with is the ability to obtain these molecules, to synthesize these molecules. And so that's where, sort of the idea behind the cache challenge would be to focus specifically on some of these ultra large libraries that are available, where we can, such as the enamine real space, where these compounds are readily available. And for this purpose, we put a compound library search functionality into Foldit. And so the concept would be that players would design the molecules using the small molecule design panel. And then once they have a molecule that they're interested in, they can then upload it to the Foldit server. The Foldit server could then turn around and query a compound library. For the cache challenge, we're actually using the Zinc Small World API, which provides access to the full 38 billion enamine real set, the full combinatorial library. We search this library. The results from that library come back. So those molecules in the enamine real set, which are similar to the query molecule that the player submitted, go back to the Foldit server, and then we can provide those to the Foldit players. And when they download those from the Foldit server, now they have a list of molecules sorted by compound similarity that they can examine, and they can now choose which of those molecules they would wish to substitute into the active site, replacing the current molecule. They can then optimize that molecule and then go on to further diversify the molecule with the small molecule design tools, and then have an iterative process where they add atoms and bonds, they look for compounds in the compound library, and then then go back and do additional modifications to that compound to further optimize the small molecule structure. In addition to modifying the small molecule structure, they can also optimize the position and placement of the ligand within in the pocket of the protein. And we provide a number of different tools to that. A sampling of such is the Wiggle tool, which does a gradient descent energy minimization. We also have what we call Shake, which is a conformational sampling. And this is sampling both the sidechain conformers as well as the internal conformers of the small molecule. There's a Move tool, which allows rigid body translation and rotation of the small molecule within the pocket. Pull, which allows players to sort of click and drag atoms in the molecule to move them to different locations. And we also have the Tweak tool, which allows players to go in and individually rotate rotatable bonds within the molecule. Throughout this process of designing molecules and optimizing molecules, sort of the primary score that the players are trying to optimize, because as Foldit is a game, the players are competing to get the high score. This score is calculated primarily based off of the Rosetta calculated energy. So this is the energy of the entire system. However, in order to favor those molecules that have the sort of the best binding in the pocket, we upweight the interaction energy. So the interaction between the protein and the ligand is upweighted within this energy calculation. But just binding energy alone, as many of you are aware, is not sufficient for a drug discovery pipeline. Additional objectives are needed. And so for this reason, Foldit has an objective panel, which allows listing of multiple different objectives that we want players to accomplish aside from just the binding energy and the energy optimization of the structure. Each of those objectives has a score value associated with it. So if you are within range of that particular objective, you get a bonus. And there's also an option to show what might be wrong about that structure. So highlight those atoms that might be contributing to a bad hydrogen bonding score, or the torsions that might be associated with number of rotatable bonds. And so for the cache challenge, a lot of those objectives were set by the traffic light scheme that was provided by the cache organizers. Molecular weights, number of rotatable bonds, presence of Payne's groups. But in addition to those provided criteria, we had additional objectives that we added in. One of these is torsional quality. So the Rosetta energy that we're using doesn't necessarily capture the internal energy of rotatable bonds as well as it could. So we supplemented this with an additional objective based off of the Rarity torsional library that will give a bonus if we have torsions that are all in a reasonable space. We also added an objective for synthetic accessibility. While strictly speaking not necessary because all of the enamine compounds are theoretically synthesizable, we found early on that adding the synthetic accessibility bonus kept players more in a space of chemicals search space that allows them to get better results from the compound library search. So as long as they're staying within that synthetic accessibility range, they have more success when they actually do submit those designed compounds to the compound library. So this leads us to the puzzle setup. So for the hit identification phase, we had 8 rounds of puzzles, 1 week each for the puzzles. And each of these different puzzles are slightly varied. So we varied which starting protein we used. We varied which starting ligand we gave them. So all of the puzzles start with a ligand that the players can then diversify and change. We also varied whether or not we allowed sidechain flexibility. However, we kept a fixed backbone throughout. And also some of the later puzzles encouraged hydrogen bonding by having objectives that gave a bonus if a number of different hydrogen bonds were made to the rest of the protein. And to focus players into the active site that we're looking at, so the RNA binding site, we provided just a trimmed structure that encompassed just the binding areas that we were interested in, listed in blue there. So from that 8 weeks of puzzle playing, those 8 rounds, we identified approximately 7,600 compounds. Now, these are not just these are not all of the compounds that the players tested and worked on. These are only the compounds that were submitted to the server. And so the ones that were submitted to the server were the top scoring ones for each player, as well as compounds that they thought were interesting and shared with us. As well as the Foldit client intermittently pulls out structures that it thinks are interesting, high scoring, and submits those as well. From these compounds, from these 7,600 compounds that we had identified, we identified 1,400 of them that were actually within the enamine real set. So the rest of the 80% of these compounds are sort of those intermediate compounds where the players had added atoms, added bonds, but weren't necessarily within the enamine real set, didn't come back from the compound library. I think part of the reason for this low number is the fact that we did not have an objective to give a bonus for being within the enamine real library. So later on in the process, we added an objective that was specifically about, is this compound within the enamine real library, or is it, some other one? And so of those 1,400 compounds, to select the ones that we would submit, one of the primary reevaluation metrics that we used was just Rosetta redocking. So we took those compounds and we took protein structure and just sort of redocked those compounds within the protein structure using Rosetta. This just standard Rosetta ligand redocking. And for this, we were looking for 2 things. One was the predicted binding energy. When we redock the molecule, do we get a good protein ligand interaction, a good predicted ligand binding energy? The other thing that we were looking for was only those molecules that were going to be redocked into about the same location, the same structure that they were designed to. So things that might have a good binding energy, but were moving in the pocket or were not where they were designed, we wanted to discard those. And so to use this, for this purpose, we looked at sort of the funnel quality or via the p-near metric, which sort of evaluates the score versus RMSD plot and sees how likely is it that we're going to be sort of in that lower left-hand corner where we have a good binding energy and a low RMSD to the design structure. So the results from the first hit identification round, 76 compounds that we submitted were actually obtained and tested. And of those, 2 of those were identified as having activity by the cache organizers. Both of these compounds were from the same puzzle, which used a 5-RLZ starting structure for the protein, used the VXG ligand as the starting ligand, also had flexible side chains, hydrogen bond objective, and the library objective. And so I show the structures of the compounds there, as well as the sort of the structure of the VXG ligand. So with those 2 successful compounds, we were invited to participate in the hit optimization round. So for the hit optimization, we had 4 rounds of puzzles, so 4 weeks of puzzles, 2 weeks with each compound. And for each compound, we did a puzzle once with the design confirmation, so in the structure that the players originally designed, and then once in the lowest energy redox structure. These also varied a little bit in sidechain flexibility, but the main addition here was that we also added a similarity objective. So since we're only interested in derivatives of these molecules, we added an objective that calculated the Tanimoto similarity to either of the 2 compounds, and gave a bonus if you were similar enough. So from the hit optimization round, we had 2,400 compounds that were identified submitted to the server. This is lower than the hit identification round, but I think that's a combination of 2 factors. One is the fact that we only had 4 rounds of puzzles as opposed to 8. The other thing is that the similarity objective would reduce the compound search space significantly. However, of those, we had a greater hit rate from the enamine real set, possibly attributable to the fact that we had the library objective on and gave a bonus to players for staying within the library. Evaluation for the hit optimization stage proceeded very similarly to the hit identification round. We're using Rosetta redocking as both the energy and also the RMSD as identification of which ones to go forward. However, we also added a secondary filter criteria discarding those compounds that had strained torsions via the Rary torsional library. So the results from the hit optimization rounds, we had 34 compounds that were tested in this. 10 of them came from the compound number 34, and 24 came from compound 40. We had 3 that were classified as active via the cache organizers, and those 3 all came from the same starting molecule, the number 40, and also all of them came from the same puzzle, the same setup. It's the redox structure and with flexible side chains. And as you can see down below, all of these molecules are very similar, using a very similar core structure, and again with a different diversification. So I think it's a little instructive to take a look at how these compounds were designed in. Again, we don't have crystal structures of these molecules yet, as far as I'm aware, but these are based off of the crystal structures of the starting molecules, as well as the player designed conformations and the redocked conformations. So on the left is the crystal structure of the starting molecule, the VXG, that all these molecules were originally derived from. And so this fits into the C-terminus of the protein, sort of in that binding pocket with tyrosine 515 at the bottom of it. And so the original crystal structure has the carboxylate that forms a hydrogen bonding network with asparagine 516 and serine 486. And then if on the right we look at the original design molecules from the hit identification phase, it kept a very similar binding mode, substituting out the carboxylate for hydroxyl to asparagine 516, but adding on this aromatic nitrile group, which is nominally hydrogen bonded to the tyrosine 515 there. Something interesting happens, though, when we redock this. So on the left, again, is the design structure, but on the right is the Rosetta redox structure. And in redocking, this molecule flips end over end. And so now it's the aromatic nitrile that is hydrogen bonding to the asparagine 516. And the hydroxyl group is now hydrogen bonding to the carbonyl between serine 485 and 486. And it's this redox structure which was the starting structure used in the successful rounds for the hit optimization. And so in hit optimization, we've identified the 3 compounds, and looking at those compounds, the designed binding modes and the redox modes of those compounds are all very similar to each other. So on the right, I only show an example of that. Again, the aromatic nitrile is hydrogen bonding to asparagine 516, but in this case, it's shifted a little bit more to pick up a nominal hydrogen bonding contact to serine 486 as well. And that hydroxyl that previously was sort of more in is now pointing off into solvent. Again, all of these are sort of predicted binding modes for, from either Rosetta docking or from the folded players. Whether these actually represent the binding modes of these compounds in reality is still an open question. And so with that, I'd like to acknowledge the people in the Myler Lab who have participated in developing the drug-it interface over the years. Also, other members of the Fold-it team that have helped out. A special acknowledgement to John Irwin at Zinc for helping with setting up the Small World API query, the funders of this project, and a particular welcome to or thanks to the Fold-it players. So we really, in the Fold-it team, we really think of the Fold-it players as co-authors, and indeed, Fold-it Players is a consortium author on all of our papers. A lot of the development of these molecules is driven by the players and what the players have designed, and we're just sort of doing the post-analysis afterwards. And I will also say that we're continuing to do drug development puzzles with Fold-it, so please feel free to download the client and participate. And we're always looking for new projects and new collaborations for targets to allow the Fold-it players to participate in development of science. Thank you.
Thank you, Rocco, for your nice talk. We already have many questions in the chat, so let me just pick one. What part of the human crowdsourcing cannot be accomplished by AI or ML, and why? Given that all parts of the objective function are computed, and it seems like a computer should be more efficient than people at optimizing towards the objective. And what can we learn from humans to make AI more effective?
Yeah, that's a good question. I mean, I think a lot of it is sort of efficiency over search space. And so, the players are able to take a look at the 3-dimensional layout of the binding pocket and come up with hypotheses as far as what might work and what might not. I think certainly it is a case that it may be in the future that, AI tools will develop to the point where they'll be potentially more efficient at certain tasks. It's certainly the case that John Jumper has actually mentioned Fold-it as an inspiration for AlphaFold. So the fact that players could do well in in Fold-it in folding proteins inspired them to work on AlphaFold. But yeah, it's so I think we're not necessarily quite there with the small molecule design. But yeah, it's is there things we can learn from the players as far as what they're looking for and what sort of techniques they use that we might inspire future AI approaches?
Right. Is there any kind of scaffold for hopping function in drug-it? And could you think of running drug-it just with an apoprotein, where you would have no known fragment available?
Yeah, so in the hit identification phase, players were not limited to sticking with the scaffold. So we give them the starting molecule just for giving them something to use a handle on. But they can potentially delete off all of those atoms, add the new atoms that they want. So they can completely rework the molecule that we give them. So from that sense, they have complete freedom to do scaffold hopping. Theoretically, you could start with an apo structure. I think there's a technical limitation in the fact that you need to provide something for the players to start with. But again, it doesn't necessarily have to be a good binding compound, as the players do have the ability to change the compound completely.
Right. So we have Ashish who would like to ask a question directly. Please, Ashish.
Yeah. Hi, Rocco. Great talk. I was just wondering two quick questions. One is, I think you have to kind of strive for a delicate balance between the ability of players and how many players you can get, right? So I was just wondering, could you make it slightly simpler and get more players to play the game? And the second quick question was, if you can other people build on top of Drugit? And if you were, what are your top features if you were to build V2 of this?
Yeah, I mean, certainly one of the primary things what we're thinking about when we're building Foldit is how do we make things, user friendly, because most of the players are indeed not experienced with biochemistry. Foldit typically is, the most advanced biochemistry that they've done. And so, yeah, that's always the thing is how do we make it easier? How do we make, the things more apparent to people? I think as far as sort of the next steps in the development, I think evaluation of the molecules is still something that's we're interested in pursuing. We initially planned to use a machine learning method in order to sort of post-rank the compounds as they came back. But that didn't really pan out. There were some technical things that didn't work quite right. So we're definitely interested in pursuing that and interested in looking at what other cache participants found successful. And it's possible that incorporating those into the Foldit scoring such that players have better feedback as far as what works and what doesn't, it would help direct them in their compound search.
Is all the code open source?
The code is not quite open source according to the open source definition. The Rosetta code is source available, especially and available for use for academic users. Because we're a game and there's some security considerations regarding people cheating, the Foldit code is not open as of yet. That's something we're kind of looking into, is whether the Foldit code can be made more available. But I think one of the limitations is, of course, is having the player base. The code itself is not necessarily the value. The value is having a player base to participate as well.
Thank you.
Thank you so much.
We have a question from Federico. He's wondering about how difficult it would be to implement an AI-based method like reinforcement learning in Drugit.
Yeah, I mean, that's certainly something we've thought about a little bit. I think there's a little bit of technical limitations at the moment, but that's a future possibility. Yeah.
And there will be one last question. There was an experimental fragment screen done on the target. And how does this experimental screen compare to computational results?
I actually haven't done that analysis for that. We didn't actually use much in the way of the fragment screening information. The extent of it was the starting structures that we used as the initial for the players. It could very well be that the players identified some of the fragment screening experimental information themselves. We we have noticed that, there are very dedicated players that will go and search through the literature for information to sort of augment what they're doing and, help them to get high scores.
Right. Was there any specific reason to choose the structure 5R-LZ? Specifically?
Not particularly, no. I mean, I think I took a look at a bunch of the structures, and I think a lot of it was the fact that it was just it was a low energy structure when we relaxed it with Rosetta, I think was the primary reason.
Right. I think we are through with the questions. So let me thank you, all the speakers of today. Thank you, great job. And thank you also to the participants for a really lively session. I really enjoyed it. And I hope we see each other at the next Cash webinar. Bye.
Thank you very much, Yvonne. Thank you, everybody, for attending today. Thanks to Bayer and BI for continuing to support Cash as our governance board members. A reminder to you all that a recording of this session will be made available on the website soon. We're also going to attempt to write a post to answer any questions that have not been answered in the Q&A session. The easiest way of making sure you know when those posts will be available is to go on to conscience.ca and click the big join the network button. That means you'll get updates, or to follow us on LinkedIn. Thank you all again for attending the Cash to Participant Highlight webinar, and we hope to see you at the next webinar.
Recording